Welcome, folks, to Chapter 16 on adaptive immunity. So previously in Chapter 15, we had talked about innate immunity, which is essentially those first responders, or that first line of defense against microbial agents, pathogens, and toxins that might introduce themselves from the environment into the host, right? And so this was the skin, snot, blood, pus, sweat, and tears. And these lines of defense were just kind of creating a barrier and clearing out generally potential hazards that were coming in from the environment, right? And so your innate immune system is going to respond to the flu virus the same as it's going to respond to Ebola. But really, we need another line of defense that's very specific, that can recognize hazards in the environment that present themselves in our bodies, and can specifically target and remove those hazards. Also, it's important that hazards that had been encountered previously can then be remembered and then targeted in secondary responses as well. And so this is the adaptive immune system, okay? And so if we think of the innate immune system as a kind of our foot soldiers that have a general response to potential invasions into our body, the adaptive immune system is more like special forces or a hitman that's really specifically targeting potential specific chemicals or specific potential pathogens, okay? And so I would recommend that you go ahead and pause the video, review any previous notes that you want to review, and then also look at Chapter 16 as to what we're going to cover, and then come back into the lecture. Okay. Welcome back. So for this lecture, we're going to kind of run it similar to Chapter 15 in that we'll cover some slides, and then I'll point you to an outside resource, and then you'll come back, and we'll kind of go back and forth between the slides and the outside resources. So let's talk a little bit about the characteristics of the adaptive immune system. We already talked about a couple of these. One, it should be specific, and it should recognize and target specific microbial agents, or what we call antigens in the environment. It should be able to be turned on and off, right? We don't want to have the immune response always being present. We want it to be responding to specific cues within the body, and then be able to turn it off as well. So that's inducibility. It should be amplifiable or clonable. So once we realize once our body, our immune system realizes that there is a potential hazard, that signal should be amplified, and we should be able to have an amplified response against that specific pathogen. And then later, turn it off. So that goes back to inducibility as well. It's key that the immune system is a response that's targeted to destroy oftentimes cells, and it cannot destroy self, right? It cannot destroy the host cells. So it should be able to differentiate between self and other. That's essential. And when you do have some autoimmune disorders or allergies, that's when this immune system is starting to respond to self rather than pathogens from the environment. And it should have memory. You shouldn't have to induce a full-on immune response every single time you get the same pathogen. You should be able to remember those pathogens or those toxins and be able to respond to them in a more targeted way in the subsequent encounters. Okay. And so the primary cells of our adaptive immune system, it will actually take us back to Chapter 15 when we talked about the components of blood, and specifically white blood cells. So the white blood cells are the ones that are really providing us with the cells that are responding in a targeted way to potential pathogens in the environment, and specifically lymphocytes. And so remember when we talked about 3 different types of lymphocytes: killer cells, T cells, and B cells? Those natural killer cells are general in their response to infection or microbial agents that come into the host, whereas T cells and B cells are much more specific. And those are the ones that we're going to focus on today. So both our T cells and our B cells are made within the bone marrow, specifically in flat bones like those of our hip and our ribs. But B cells mature there, and then T cells actually need to go through another series of tests, which we're going to be talking about later on in the lecture. And they go to what's called the thymus. The thymus is around our heart area, and it's part of our lymphatic system, which we'll talk about in a minute. And essentially, those cells go to the thymus. They're exposed to a series of different antigens and a series of different tests before they are matured and released into the body. Okay. And so the two types of cells, T cells and B cells, they work together, but generally speaking, B cells work within the lymphatic system, which is essentially a system that allows us to search through the blood, specifically plasma, a liquid that comes off of the plasma of the blood, and search through it and see if there are potential hazards that are coming in from the environment. Whereas the main job of T cells is actually to search through the cells of the body and detect cells that have actually become infected. So a pathogen has come in and has been missed by the B cells in the lymphatic system, and then T cells are then there to kind of see that cell has been infected, or can recognize that cell has been infected, or in the case of cancerous cells, has become cancerous and can eliminate it. Okay, and so those two types, cell-mediated immune responses, is related to kind of perusing the cells of the body and indicating or targeting cells that have become infected, whereas antibody immune response is related more to B cells, and that is them searching through what's called lymph that comes off of the plasma of the blood to look for incoming pathogens. Although T cells and B cells do work together, so these two processes are not mutually exclusive. Okay. Here's just kind of an overview of what's going on with host defense.
Host defenses consist of two categories: the innate or non-specific defenses and the adaptive or specific defenses. The human body has many innate defenses to prevent disease. These include the first-line defenses such as the skin and mucous membranes, phagocytosis of microorganisms, inflammation, and the complement system. The human body has two main divisions of adaptive defense: humoral immunity and cell-mediated immunity. Antigen-presenting cells aid in the activation of these.
I just want to indicate that the humoral immunity is related to antibodies, which is this antibody immune response related to B lymphocytes, just to make that connection, because sometimes we use that different name.
Inflammation, and the body has two main divisions of adaptive defense: humoral immunity and cell-mediated immunity. Antigen-presenting cells aid in the activation of these defenses. A humoral immune response consists of the activity of B cells and the antibodies they produce. Cell-mediated immunity is a host defense used to fight intracellular pathogens and abnormal body cells, such as cancer cells. The protagonists of cell-mediated immunity are T lymphocytes, which can be divided into two groups: helper T cells and cytotoxic T cells. Helper T cells secrete cytokines to activate other cells of the immune system, while cytotoxic T cells attack infected cells directly. The innate and adaptive components of the immune system work together to carry out host defenses.
Okay. So we're going to start off our discussion of the adaptive immune system by talking about the lymphatic system, and then we'll go into immune responses, and then we'll talk about how do we actually, develop immunity over time and in various ways. Okay. So let's start with the lymphatic system. What I want you to do is pause the video and go into the first resource, which is a crash course video about the lymphatic system, and then come back into the lecture video. Okay, so as you saw in your crash course resource, the lymphatic system is essentially taking parts of the plasma, or plasma, out of the blood and searching through it to see if there are potential invaders. And so the lymphatic system is a series of tissues and vessels that are just throughout our whole entire body and allow us to process that blood and see what's coming in with it. And so this colorless fluid that comes off of the plasma, it has within it many things. It has different gases, it has metabolites, but it also may have incoming microbial agents and pathogens. And so your lymphatic system is specifically focusing on, searching through that liquid and looking for the presence of potential pathogens. And this is why our lymph nodes swell when we're sick, in our neck, for example, and in other areas of the body, because you're actually having recruitment of many white blood cells into that lymph in those areas to process and destroy pathogens that are coming in. Okay. So you can see here our lymphatic system. We talked about the thymus, which is in this area near the heart, but also part of our lymphatic system are our tonsils, our adenoids, and then what we call malts, or mucus-associated lymphoid tissues. And those are the mucosal regions of the urogenital tract, the respiratory tract, and digestive tract. Okay, and so in addition to capillaries, you'll also have these lymphatic vessels that go through these areas and bring the various immune cells to those areas to respond to and check for pathogens. Or what is it that the immune cells that are present within the lymphatic system, what is it that they're patrolling for? And the answer is antigens. Okay, and an antigen is a general term for foreign bodies that are coming into the body, foreign molecules, foreign particles that are coming into the body. Okay, so this could be bacterial agents, it could be toxins, it could be various types of allergens that are coming into the body that the body is rejecting. And then there's specific regions within the antigen that will actually be identified by the immune system. And oftentimes one single antigen will actually have many different, what we call epitopes, or regions that are actually recognized by many different immune cells. And so just to kind of put in perspective the terminology, and we're going to talk more about that. Okay. And so when we're talking about antigens, our own cells have what we call autoantigens on them. They display proteins from within them. This actually actively kind of take proteins that are made within the cell and display them on the outside of the cell. And this is kind of like presenting identification at the airport, or showing your bags at the airport, just kind of displaying what's already inside of those cells. Those autoantigens are markers of self. Okay, and so the immune system generally, if working properly, does not respond to autoantigens, whereas you have various types of foreign antigens that are markers of non-self, and that's what your body is going to respond to. Okay, so let's talk a little bit more about those. First, we're going to talk about the anatomy of an antigen, and then we're going to move into the different types of antigens, so we'll get back to autoantigens and foreign antigens. Okay, so as we had said, antigens are these foreign particles that are coming into the body, and whatever you have different particles that might come in. It might be a full bacterial cell, and then it's presenting on its the outside of the cell with the various components of the cell wall and the cell membrane, maybe flagella, pili, right? And all of those different kind of parts of that foreign body, of that bacteria, can be recognized as parts of that antigen. And the specific shapes or the specific units that are actually recognized by the immune system are called epitopes. And epitopes, essentially, oftentimes they're proteins, and oftentimes they're large so that they can actually be recognized in terms of essentially being more efficient epitopes. So like we said, proteins tend to be more efficient or more recognizable than other macromolecules, although other macromolecules can be epitopes as well. The larger the epitope, the easier it is to bind and recognize. And the more complexity that an epitope has, the easier it is to identify whether that is self or non-self. So for example, carbohydrates are very general. They're very kind of just repeating subunits of the same glucose molecule. So that doesn't provide a lot of complexity to kind of identify or narrow down whether this is self or non-self. Whereas proteins tend to be more complex and more efficient as epitopes in terms of recognition by the immune system. So now that we kind of understand how an antigen kind of is and what parts of it are recognized, let's talk about different types of antigens. So we had mentioned that our own cells, which you see here, they're always displaying their own proteins, okay? And this is an important part of the immune system and the development of the immune system. So we're going to talk more about how various cells in the immune system develop. And essentially part of that process that they go through is recognizing the various proteins that us as human hosts tend to make, right? And when those proteins are presented on the outside of the cell, that indicates to the immune system that this cell is functioning correctly. It is self versus non-self. And it should be left alone. It's not going to elicit immune response, and they just kind of move on from there. Whereas if a cell is infected and it's presenting then strange proteins, like it's infected by a virus and the proteins that it's presenting on its cell membrane are viral proteins, then the immune system is going to be able to recognize that and respond to that. Also, if you have microbial agents, toxins, or pathogens that are coming into the cell that themselves, they are just showing antigens, on their own surface, right? So flagella, different types of proteins that are non-self, then those can be targeted and responded to. And so these three types of antigens, we call them autoantigens, that's self-antigens, endoantigens or endogenous antigens, and these are antigens that are coming from the inside of an infected or affected, because also cancerous cells can present endogenous antigens coming from inside of an infected cell. Exogenous antigens are actually particles or pathogens that are coming in from the environment that they themselves are the antigen. They are the ones that are kind of non-self or presenting as non-self, okay? And the immune system will respond to these in various ways. And essentially the cell-mediated response is going to focus on, what antigens are being presented on the outside of our cells, whereas your antibody response is going to focus on these kind of invading particles from the outside. So here is the cell-mediated immune response, and this is when your immune system detects cells that are presenting non-self antigens.
Cell-mediated immunity is a host defense used to fight intracellular pathogens and abnormal body cells, such as cancer cells. The protagonists of cell-mediated immunity are T lymphocytes, which can be divided into two groups: helper T cells and cytotoxic T cells. Helper T cells secrete cytokines to activate other cells of the immune system, while cytotoxic T cells attack infected cells directly.
Okay, so if we remember the T cells, we had mentioned they're made in the bone marrow, they're produced in the bone marrow, but they actually mature in the thymus. And so the thymus being part of the lymphatic system, and it right here in this area around the heart, and interestingly, it's an organ in which the T cells go to, they migrate to. There's essentially chemical signals that are sent out, and these immature T cells actually migrate to the thymus. And in there, the thymus is actually producing all of the proteins that are normally produced by the human body, even proteins that the thymus itself is not utilizing. And it's presenting those proteins to the T cells. And any T cells that might have a complex on its cytoplasmic membrane that responds or kind of would mount an immune system towards self is actually apoptosis. And apoptosis means essentially it self-destructs. And so within the thymus, these T cells are developing exposure to potential antigens, but they're also developing exposure to proteins that indicate self. And so that allows for the body to kind of clear out any T cells that might mount an immune response towards self and actually allow for other T cells to develop exposure to potential antigens. Okay. And so, in terms of recognition, this is a distinction between T cells and B cells. The T cells are not recognize the epitope itself. It actually recognizes the complex that's created with the MHC proteins that are on the outside of the cytoplasmic membrane. And when an abnormal complex is detected, that's when the immune system is recruited via the release of cytokines, which are chemical that essentially say we need more white blood cells in this area, and then the destruction of that cell essentially. So here you see one of the cells of the body that's presenting an antigen on one of these MHC protein complexes, and then the T cell is recognizing that and will respond to it. So there's kind of three different types of T cells. Cytotoxic T cells, which are the ones that actually directly kill the cell that's displaying that abnormal MHC complex. Helper T cells, which help regulate the various cells of the immune system, and also release cytokines, which essentially bring in more white blood cells into the area. Both of these two types of cells can continue to kind of display these antigens kind of over the long term on their cell membrane. And that allows for memory within the immune system, which we talked about being really important. And then also regulatory T cells. So if you can think of helper T cells as kind of the on switch, they release cytokines and they bring in the backup and reinforcement of the immune system. Regulatory T cells are essentially the off switch. Now, it's not exactly that simple, but they release different types of cytokines that essentially kind of inhibit an immune response once the threat has been abated. Okay, so that's essentially our cell-mediated immune response in general. The second part of the adaptive immune system is a very targeted response to antigens that are coming in to the body from the external, right? And so that is going to be kind of curated mostly by what we call B cells in relation to the antibodies that they produce. So here's an overview of what can be called the humoral immunity or antibody immune response. There's various names, but essentially related specifically to antibodies that recognize antigens from the external environment in the host.
The adaptive immune response is divided into two categories, humoral immunity and cell-mediated immunity. A humoral immune response consists of the activity of B cells and the antibodies they produce. The humoral immune system experiences periods of ramping up and gradual diminishing in response to a foreign invader. After the first exposure to an antigen, there's a lag period in which the exposed person's serum does not contain any detectable antibodies. After several days, the concentration of antibodies slowly rises, followed by a gradual decline. This response is called a primary response.
Okay, so let's talk more about the B cells of the antibody immune response. And so B cells essentially are white blood cells that are able to recognize epitopes on antigens. So remember, the antigen is the foreign body, and the epitopes are those specific regions that are recognizable by the immune cells on that foreign body. And you have multiple potential epitopes on one antibody, but the B cells, as they mature, they only develop the ability to interact with one specific epitope. So they have proteins that they kind of show on their exterior, on their plasma membrane, called B cell receptors, and those are specific to an individual epitope. one B cell is going to have many of those receptors, up to hundreds of thousands of those receptors, but all of them will be exactly the same, and they'll essentially be seeking out that one epitope. And therefore, you will likely have many different B cells that could recognize the same antigen because they're actually responding to different epitopes within or on that antigen. Okay. And so these B cells are kind of, roaming through your lymphatic system and looking for these potential antigens. And when the antigen is recognized, so when you have the binding of the epitope that's very specific to that B cell with its receptor, then there's an immune response that's elicited. So you have cytokines that are released to bring in other white blood cells, but also the B cells will produce antibodies. And so an antibody is actually very similar to, it's essentially this same B cell receptor, but without this transmembrane part. So you see how this looks like an antibody, right? And so it's essentially the same kind of protein complex, but without this transmembrane piece. And those are kind of produced throughout the cell and then released. And it's not just one like in this cartoon. Each cell will release hundreds and thousands of these into the lymph and into the bloodstream to elicit a response to that specific antigen that has been recognized. And so here you can see how the antibody looks very similar to those B cell receptors. And so what is the goal? What is the purpose of these antibodies? Well, they do a number of different things. One, they bind to the foreign body, right? The antibodies themselves, separate from the cell that produced them, bind to the foreign body. And so that allows for the activation of the immune responses. So immune responses like the complement system, inflammation, recruitment of white blood cells, so on and so forth. Also, it can neutralize the threat from that pathogen just by the fact that it's bound up, right? If you have these antibodies, here's another image here. These are the antibodies. This is the antigen or the threat. And once you bind up those antibodies to the antigen, sometimes it just doesn't have access to receptors to actually infect cells more. Opsonization is that binding of the antibodies to the antigen is a flag, and it actually tags that antigen for destruction. Agglutination is when you have antibodies actually connect with one another and the pathogen ties them up and kind of keeps them separate from the cells of the body until they're digested by macrophages. And then also recruitment of macrophages to actually take out and kill that specific pathogen. So natural killer cells, for example, coming into the site and digesting that pathogen or that threat through phagocytosis. So you can also see that here, where we have our antibodies that are associating with the antigens, either to flag them, neutralize them, or agglutinate them, and then the macrophages are recruited to that area to actually destroy those antigens. Then actually B cells, some of the B cells will actually present parts of those antigens on their plasma membrane, and that will provide a long-term memory of that antigen within the body. And that way, if there is a secondary infection of that specific microbial agent, pathogen, then the body will be able to mount a quick and effective immune response to that. And this is what a secondary response would look like.
During the primary response, some activated B cells differentiate into long-lived memory cells. If these cells become stimulated by the original antigen, even years later, they rapidly produce antibody-secreting plasma cells. This faster, stronger response is called a secondary immune response, a memory response, or an anamnestic response.
Okay, so that concludes our summary of the cell-mediated immunity and also antibody immunity, also called humoral immunity. And so you can go ahead and pause the video, jump into the 2 crash course links that are below on the immune system, and use those to review the content that we just covered. Okay, welcome back. So we can see from our discussion of the immune system is that when we are presented with an antigen, our body is actually able to respond via an immune response, but then actually remember that antigen for potential later infections. So the question arises from a medical perspective is there a way to kind of bypass that initial illness or infection and actually develop some sort of immunity outside of that? And so that brings us to the discussion of acquired immunity. And so there's 2 different ways that we can acquire immunity: natural acquisition, which is part of what we discussed already, as well as artificial acquisition. And then within that, there's kind of subcategories, whether that's active or passive. And so we're going to talk about those 4 categories of the kind of acquired immunity next. Okay, so naturally acquired immunity is what we talked about already, is when our body is actually exposed to an antigen and it actually responds via an immune response and then produces antibodies that can then mediate and kind of help improve that secondary response's efficiency if that pathogen is then experienced later on in an organism's life. But especially in the case of children, you might not have that capacity. That immune system is still developing, and they might not have the capacity to actually respond efficiently to pathogens or antigens. And so we can, as children, receive antibodies from our mother, either prenatally or via breast milk. And so that allows for babies to kind of gather that immunity that they need as their immune system is still developing. There's also mechanisms for artificially acquiring immunity. And so the primary mechanism is vaccines. Essentially, what you're doing is introducing an inactive kind of particle or full antigen into the body of the host, and then that immune response is mounted, and then you kind of have that memory then afterwards. There's another way for receiving artificial immunity, and that's through passive mechanisms. And essentially that's what we call, the antidote. And so what that is if there is a toxin or an infection that is life-threatening, and immediately you need to take antigens from another host and put them into the newly infected host. That could be in animals that are being used to produce antibodies, or from other human hosts then those antibodies are transferred into the infected individual, and that it allows them to mitigate the response to that illness. And so, for example, antidotes to various types of venom, viruses like rabies or Ebola as well, more recently, are ways in which you can acquire passively artificial immunity. There's this video here that gives us an overview of kind of what we talked about in terms of the immune system overall. There's another video that's the last video in the links that are provided in the description that also gives another overview. There's some detail in both of these videos that you won't necessarily need to know, but do watch them so that you can see how all of these different components of both the innate and adaptive immunity work together to protect us from pathogens or potentially hazardous microbial agents in the environment.
This patient's cold is caused by rhinoviruses, shown here in red, that entered her nasal passages and escaped the first-line innate defenses such as mucus and cilia. Some of these rhinoviruses entered the respiratory epithelial cells where they replicated, releasing more rhinoviruses into the patient's nasal passages and tissue fluids. In the tissue fluids, cells of the second line of innate defense, such as this dendritic cell, carry out surveillance for anything that appears foreign. Dendritic cells engulf the rhinovirus within an endosome. The endosome fuses with a lysosome, whose enzymes digest the rhinovirus. The resulting rhinovirus fragments are called epitopes. The dendritic cell then attaches the rhinovirus epitopes to presentation molecules called MHC2. This complex folds outward so that it sits on the outside of the dendritic cell membrane. The dendritic cell then enters a lymphatic vessel and travels to a nearby lymph node. Here, the dendritic cell, part of the innate defenses, encounters a helper T cell, which is part of the adaptive defenses. The T cell receptor, or TCR, of this helper T cell recognizes the rhinovirus epitope, while the CD4 glycoprotein associated with the TCR recognizes the MHC2. The dendritic cell then stimulates activation of this helper T cell. Adaptive defenses help to control the current infection, but also provide immunity against future infections by the same rhinovirus. They are carried out by two types of lymphocytes, B cells and T cells. Immature B cells conduct surveillance in the lymph nodes, using B cell receptors, or BCR, to recognize only one specific epitope. The BCR on the left does not recognize the rhinovirus, but the BCR on the right does, and this B cell is selected for activation in a step called clonal selection. The selected B cell takes in the rhinovirus, degrades it, and presents the rhinovirus epitope with its MHC2. Our mature helper T cell recognizes the rhinovirus epitope with its TCR and the MHC2 with its CD4. Then the helper T cell secretes chemical messengers known as cytokines that activate the B cell. The B cell proliferates and differentiates in a process called clonal expansion, resulting in two types of cell. Memory B cells carry the memory of this pathogen for many years. Plasma cells secrete antibodies that conduct surveillance for more rhinoviruses, grouping them together so they are easy targets for phagocytes. Antibodies in the blood and tissue fluids seek out free rhinoviruses. The antibodies neutralize the rhinovirus by binding to its surface molecules, thus preventing attachment to host cells. Antibodies are humoral defenses that target free pathogens. But what about the rhinoviruses that are already inside our patient's cells? Another type of adaptive defense cell, called a cytotoxic T cell, can locate and destroy rhinovirus-infected cells. It provides cell-mediated or cellular immunity. Let's return to our patient's lymph node to see how cytotoxic T cells become activated. The T cell receptor, or TCR, of this particular cytotoxic T cell is genetically programmed to recognize the rhinovirus epitope when presented by the dendritic cell. This time, the epitope is presented differently, with an MHC1, which is recognized by a CD8 glycoprotein on the cytotoxic T cell. With the help of cytokines from a nearby helper T cell, the
Okay, so it seems to be an issue with loading the end of this video, but essentially the cytokines then go out and elicit an immune response. So that's our discussion of adaptive immunity. I would recommend that you review your notes and review the resources that are presented. And there's not a specific Jamboard for this lecture, although you will have interacted with the Jamboard this week in chapter 15. So that's that for today, and I'll see you in the next lecture.